My name is Patrick Vanderboom. I'm from Mayo Clinic in Rochester. And today I'll be talking about some of the research that we're doing. We are trying to identify extracellular vesicle-based markers of neurodegenerative disease, specifically within ALS. I'll start with a bit of background in the project. So, quick overview. So we're going to cover some background in the EV isolation method that we're using. It's a hot topic these days. Then we're going to talk about ALS and somewhat just kind of a high-level overview of the disorder, and then talk about the project that we're working on, and I'm here to talk about today, which is identifying EV-based ALS-specific markers of disease pathology. We'll talk about the discovery experiment we did and some follow-up targeted validation. So what are extracellular vesicles, or EVs? There's three different categories of EVs. It can be divided into exosomes, microvesicles, and apoptotic bodies. So exosomes are formed by inward budding of the early endosome, which then matures to form the multivesicular body, which can either fuse with the plasma membrane to degrade those proteins, or fuse with the plasma membrane to release those inner contents as exosomes into the extracellular space. Microvesicles are distinct from exosomes because they bleb directly from the plasma membrane. And they're typically distinguished by size. So exosomes are thought to exist in the range of 20 to 120 nanometers in diameter, whereas microvesicles can be much larger. But there is some overlap. So we typically refer to them as small EVs or large EVs, where small EVs are enriched for the exosomal population and large EVs are enriched for microvesicles or potentially apoptotic bodies. So we use size exclusion chromatography to isolate EVs from plasma or any complex biological matrix. So this particular figure is showing just the scale protein abundance of several EV marker proteins as they elute off an SEC column. So the thing about SEC is it separates by size. So large molecules are going to elute from the column first, whereas small soluble proteins are held back as they interact with the pores in the resin. So we can see here that if we, in this particular experiment, we took each one of the fractions from the SEC column, we palleted it individually with ultracentrifugation, and then we plotted the protein intensity, and you can see that you can construct a pretty nice chromatographic peak off the column. Along with those EVs come a little bit of plasma protein background, so that you can see that these are high molecular weight proteins like ApoB, IgM, fibrinogen, but you can kind of resolve them chromatographically. So you can then use this information, like in terms of what proteins are eluting in the same region as the EV marker proteins, to identify with higher confidence which proteins are actually from the extracellular vesicles. So just to be clear, we don't always do this experiment. We don't fractionate in each individual sample, but this type of experiment could be done to figure out where EVs are eluting from the column. Then you combine all those fractions into one, and pallet it, and then process it as one individual sample. But you do have the capability of essentially doing chromatography on EVs from plasma. So once we kind of honed this method in, we did some analytical characterization. So in this particular experiment, I'm just describing some linearity precision studies that we did. So we took neat human plasma and we diluted it at several twofold dilutions using exosome depleted fetal bovine serum. We used 5 different SEC columns for this experiment. So then across the columns at each dilution level, we could calculate imprecision or CVs. And then within each column, we could calculate the linearity in the form of R squared here. And what we found from this particular experiment is that it's pretty good. So we have good linearity for, as represented by four representative EV marker proteins here. But in the entire data set, we had identified over 850 proteins with a R squared of over 0.9, which is pretty good. And I'm not displaying it here, but the mean imprecision, or CV, at each dilution level was less than 25%. So for a label-free workflow, this is pretty good, opening up a lot of opportunities. So when we developed this method, we applied it, of course, to a couple of the projects that we've worked on over the years, and I just want to highlight a couple at a very high level to illustrate some of the things that you can do with this. So one of the first things that we applied this to was, at the time, I was in an endocrine research unit, and we were interested in studying the response to exercise, and specifically we knew that there was a release of extracellular vesicles after intense aerobic exercise, but we needed to confirm that and try to identify specific markers within the EVs that represent certain tissues within the body that are responding to exercise. So this is the same volcano plot in each facet. It's just overlaid with some candidate tissue-specific markers that we identified from some cell culture experiments. Slightly more clinically relevant problem that we were studying is, we're a big myeloma center at Mayo, and one of the things that we wanted to look at was these patients with multiple myeloma, they have a precursor disease called monoclonal gammopathy of undetermined significance. And monitoring the transition from MGUS to myeloma is problematic. It's hard to do. You have to catch the patients before they end up with end organ damage. So we wanted to see if there's some potential novel markers of disease burden in these patients that we could identify from EVs. So another thing I want to highlight from this experiment is that we took the EVs and pelleted them two different ways. So we can pellet the large EVs at a lower centrifugation speed, take that supernate and pellet the small EVs at a higher centrifugation speed. In both populations in this experiment, we identified transferrin receptor as upregulated in the myeloma group, which is intriguing to us. And then further, if you took elevated transferrin receptor protein intensity from the large EVs, it was significantly associated with a reduced survival in this cohort. So it's something we want to follow up on, but it just kind of illustrates the fact that these different populations of EVs can have distinct biological meaning, and the proteins that we identify in both fractions are distinct. Relatively distinct, of course, a lot of overlap. So when I became a lab director at Mayo Clinic, one of the things I wanted to do was follow through. I wanted to take this method into the clinical laboratory to try to identify those next generation biomarkers that we could actually implement into the clinic to just provide better care to the patients that we serve. So one of my colleagues and my friend at Mayo Clinic is a neurologist who specializes in neuromuscular disorders. He was doing a study on amyotrophic lateral sclerosis, or ALS. And together we decided that we would apply this method to a cohort of patients that he had to see if we could identify some novel markers. So what is ALS? Amyotrophic lateral sclerosis, or ALS, is also known as Lou Gehrig's disease. It was named after the famous Yankees left-handed slugger who suffered personally from the disorder and brought a lot of awareness to the disease. So ALS is a progressive neurodegenerative disease affecting the central and peripheral nervous system. It leads to a loss of voluntary muscle control, atrophy, and eventual paralysis. 10% of the cases are familial, involving mutations in several proteins, including TDP43. The diagnosis of ALS is primarily clinical. Patients typically present with signs of upper and lower motor neuron degeneration and a progressive spread of symptoms in the absence of an alternative explanation, such as paraneoplastic syndrome or B12 deficiency. EMG typically shows denervation and fasciculations, so like spontaneous muscle contractions. And there are some laboratory tests that can help, like plasma neurofilament light chain. So there's a strong correlation with neuron death. However, it's not specific to ALS, so its use in the diagnostic setting is of limited utility. So there's several unmet diagnostic needs in ALS. There are currently no specific markers for ALS. Distinguishing ALS from mimics in the early part of the disease is problematic, and it affects treatment. You can imagine if you were suffering from something where you had progressive weakness in a limb, and you were wondering if you had ALS, that can cause a lot of distress. So it'd be nice for both patients with and without ALS to distinguish between these groups. There's also a need for more prognostic and treatment response markers to facilitate clinical trials, because there's a lot of room for improvement in this disorder. So with these limitations in mind, we set forward to identify some novel markers, EV-based markers in ALS. So we isolated EVs from CSF from 28 ALS cases and 21 controls with various neurodegenerative diseases, or just neurologic diseases. So the workflow looks a little bit like this. So we take we take the CSF, which is 0.5 mils. We pellet debris at about 2,000 G for 10 minutes. Then we isolate the EVs using SEC, and then we pellet the large EVs at 20,000 times G and the small EVs at 100,000 times G. So in this experiment, we're specifically focusing on the small EVs. So we took that forward, we digested it, and we worked with our friends at Thermo to analyze it at an Orbitrap Astral with DIA. And then we found some candidate markers, and we followed up with some targeted validation at Stellar. So for the discovery experiment, we used Vanquish Neo coupled to an Orbitrap Astral, as I just mentioned. In terms of the separation, we used 25 centimeter, 1.7 micron ion optics column, operated with a little bit of heat, a pretty standard gradient, flow rate of 300 nanoliters per minute, and a total method runtime of 37 minutes. The Astral was operated in full MS mode. It was 240K with 500% AGC, scanning from 380 to 980 M/Z with 100 millisecond injection times. For the DIA scans, we had 200 windows that were 3 Daltons wide with 80,000 resolving power in the Astral analyzer, 25% HCD with a 500% AGC and a 7 millisecond injection time, so really fast. We had no idea what EVs were going to look like on an Astral. We had never done it. We typically run it through our standard workflow, which is on a Tribrid or an Explorers 480. So we wanted to start with comparing it to something that we're really familiar with. So we ran 3 technical replicate injections of a HeLa sample and a pooled EV sample that we had prepared before we sent the samples out. So in this analysis, upon the 3 replicate injections, we found that we had quantified 8,560 total proteins from the HeLa with a median CV of 4.29%. In the EV samples, we quantified just under 2,000, 1,919 total proteins with a median CV of 11.6%. So you can see the sample types are a lot different. Our samples are much more sparse than you'd find from a cell lysate, representing some technical challenges. But just to level set, this number of 1,919 from quantified proteins was jaw-dropping for me because we had typically not even gone over 500 when we're using our typical workflows. This is a huge leap forward, very exciting. In terms of the actual results from the discovery experiment, we had 2 different control groups. So we had normal pressure hydrocephalus or NPH controls, and then additional set of controls with non-NPH, so other neurological disorders. And when we were selecting our candidates, we wanted to make sure that we were selecting the best ones. We required that they were differentially expressed relative to both control groups. But in total, we identified 5,771 total proteins. When you require 2 peptides as a minimum, almost 400 of them were differentially expressed. Most of them went up in the ALS group. We did some further clustering, and by and large, all samples, most samples clustered within their respective groups. So we were super excited when we got this data. I've been doing this for a while. I've never seen anything like it in terms of coverage. So it was a completely new paradigm for us. So we went into our typical workflow of targeted validation. So generate some candidates, follow through with the targeted validation. We're working on a mass spec that we had at Mayo, is at Explorers 480, which is an awesome instrument in its own right. But I had kind of suspected the fact that we were identifying so many more proteins on the Astral might put us in this position where we were trying to validate candidates that were present at just very low abundance. We might have trouble identifying them with, by our traditional means. So we developed a PRM assay for 10 total proteins, 2 peptide targets each. So we synthesized 20 total stable and stable peptides. Here's 2 representative proteins, called protein A and protein B. So we're seeing the trends that we expected to see based on the validation data. But if you look at all the protein targets that we monitored, you can see from all the blue in this heat map that the blue is representing no signal. So most of the peptides, we just weren't able to get any signal from with our PRM method. So this kind of confirmed my suspicions that we were in this kind of zone of, promising candidates, but challenging sensitivity. So at that point, I reached out to Carrie Hassel's group and said, hey, can you help me? I don't have a Stellar, but I'm pretty sure we're going to need one because it's faster, it's very sensitive, and we wanted to validate some of these targets. Someone on my team and I went out to San Jose to visit with Carrie's group. We learned a lot about the Stellar during that visit. One of the great things about it is that it's got some software that's integrated that can really help streamline the discovery to targeted transition. So we played around with that quite a bit, and it was impressive. So we had identified our candidates. We selected made our short list of protein targets, and then when we went out there, we used PRM Conductor to kind of refine the list. We experimented with some MS3, which was really cool, and eventually, kind of with the theme of today's talk, our hope is to get this streamlined to the point where we're not spending weeks, or potentially months developing these targeted assays. We want to go from discovery directly to, within a week or so, a targeted method to really push these candidates through at a faster rate. One thing I wanted to highlight from some of those experiments was our experimentation with MSN, or MS to the 3. So here's one peptide. On the left, we're looking at the MS2 level quantification, or XICs. If you look at sample 28 here, here's what it looks like with MS2. You can see there's quite a bit of noise in the background. But if you look at that same peptide when we acquired it with MS to the 3, it just cleans up the background. It's almost like high resolution. So this is really exciting and promising, and something that we want to play with more when we have a little bit more time. We also did, because I was so concerned about these sensitivity challenges that we were up against, we did a lot of characterization of the limit of detection and quantification. It's not, for some reason the slide isn't coming across clear, but we were, we had low atomodal sensitivity for many of the peptides. So we were pretty impressed with that. We also took, sorry, I should take one step back, just to clarify that when we're running these samples that I'm showing here and the ones that we showed from the 480, they're actually residual digest from that original discovery experiment. So it's really just technically validating that the assay is working and we're seeing the trends that we expected to see. So this is an additional 10 total cases from that original discovery experiment, where we ran the residual digest on the Stellar with our targeted peptides. Again, we're seeing the trends that we expected to see. We're seeing, I would call, much more robust signal, many more points across the peak, and good signal to noise. So this is promising. And then when we ran this analysis on all of them, we're getting more signal from more of the peptides. For the peptide targets that we're monitoring had complete discrimination between the cases and controls. And then 8 of the peptides had an AUC of 6 peptides had an AUC greater than 0.8. So this is really promising. You can kind of see too that some of the variability in the sample. So we had one sample who was just super high for everything. So we are seeing a lot of the targets that we weren't seeing before, seeing some variability amongst the patients, but we're moving in the right direction. So at this point, I was getting excited to add a few more peptides to the list because I thought, it just gives us a better chance to kind of move forward with this. And we identified 10 additional cases, 10 controls, actually 20 controls, 10 Alzheimer's disease controls, 10 other neurological disorder controls, and went through with external validation. So this is what it looked like. So we had a total of 20 controls, 10 cases, AUCs. 4 peptides were validated with an AUC of greater than 0.8. This isn't exactly what I was hoping to see. Although it's definitely a step in the right direction. I think what I had done is potentially shot myself in the foot because I added so many more targets to the list before I did this analysis. We weren't really scanning these as long as we should have, so we weren't filling as much. I think we can improve on this a lot. But even this data alone, I think, is pretty promising. The fact that we're doing pretty crazy stuff. We're identifying EV-based markers from CSF, a limited amount of CSF, and actually brought that through validation. So I think our main challenges still remain sensitivity, but I think that we have the direction that we need to go in from here. And I think we'll actually get there to hopefully more towards that complete discrimination between some of these markers. But of course, as somebody that works in a clinical lab, when things don't go exactly how you thought they'd go, you start troubleshooting and you figure out, where the problem is. So we started to do some studies to really characterize how the assay is performing in CSF. So all the stuff I showed you before was from plasma. But we really dug in and we figured out, we characterized the imprecision according to CLSI's guidelines of the CSF method. So we did a 3x5 experiment. We used a pooled CSF sample. Every day we extracted 5 replicates across 5 columns. And we pelleted the EVs, froze them, and digested them all in a batch on the last day. We did 2 different methods. Our original method is displayed on top. And then a completely different method using a different MS-friendly protease max in this case. In both methods, the median CV was about 65. So it's not, that less than 25 we saw with before with plasma. But there's a couple of caveats to this because we're using a quarter of the material we started with from plasma, and EVs are known to be less abundant in CSF than they are from plasma too. So I think we have much less total material. Coming at this with 2 different methods, I think this data kind of indicates, it's just showing us what we expected to see. We're pushing the limits really hard. So I think that the imprecision, although, and this isn't what I ideally hope to show, it's truly representative of the variation of this process. So we know now that when we're going to identify markers, we can really quantify like what certainty, what fold change we need to have to, identify those differences with certainty, which I think is helpful. It's, incremental science moving things forward. One thing I failed to mention is, there's 2 different types of imprecision you can do within lab and within run. If you've ever done this for your, assays in your lab, within run here would indicate like 1 day of isolation, and within lab would indicate like the several days of isolation. You're kind of using a model to parse out the 2 different factors that contribute to the variability. One thing that we looked at is we kind of rank ordered these proteins in terms of their imprecision, and then overlaid some EV marker proteins. So we can see that some of the EV markers are present, and quantified with less imprecision. And similarly on the bottom here, we're looking at the imprecision from some neuron-specific markers. So I think that, granted the median imprecision is a little bit higher. I think this is realistic, and it's still exciting. We know where we need to be to kind of continue to move this forward. We can optimize the method, both pre-analytical and analytical. But I think we're still in a good position to find some potential novel markers. We kind of have. So we've followed through with the validation in an independent cohort. Although we didn't see complete discrimination, we're definitely moving in the right direction. We have some markers that are relatively specific to ALS. So in conclusion, in the discovery experiment, we identified almost 6,000 proteins from the small EVs in this experiment. Depending on how many peptides you required, almost 500 were differentially regulated. In the targeted analysis on the 480, it showed us those expected trends. We identified that there might be some sensitivity limitations with the method that we were using. The Stellar definitely seems to improve that. And it fits really well into this discovery to validation pipeline that we want to do, based on the integration of the conveniences afforded by the software. In terms of technical characterization, the median CVs for the untargeted workflow are a little bit high, but given the low sample input, the total protein content and the complexity of this workflow, if you really think what we're doing from end to end, I think this precision might reflect, what it ultimately is. The other factor is, we acquired that precision data on a Exploris 480. I think if you were to use something like a Stellar or Astral, we know that we've got more sensitivity. Does that decrease the imprecision? Are we just too low? Is that signal too variable at those levels? So I hope I can leave you with, I think we're at an exciting time where we can actually identify markers that, a few years ago we had no chance of seeing. But now we're not only identifying them, we're moving them through the validation pipeline. And and that's